Go ahead and get started?
Yeah. Go ahead and get started. Everyone's been introducing themselves, just really a brief introduction and then.
Yeah, of course.
Yeah.
So unlike Ram, I don't know all of you guys. I've met a few, of course. So I'd love to see your faces. So if you're comfortable putting your video on, so I can associate some faces with names, that would be great. Don't be embarrassed of your background. As you can tell, I'm working out of my basement, so it can't be worse than mine. I'm Ben Prosser. I'm an assistant professor at the University of Pennsylvania in the School of Medicine. I'm in the Department of Physiology, Pennsylvania Muscle Institute, and the Cardiovascular Institute. And I am a muscle biologist by training. And so my lab works on muscle mechanics and muscle mechanobiology. But I will be sure not to impose too much of my muscle bias into this talk. This will be about more generalized principles of nuclear mechanobiology that are applicable to all cell types. Although I'll still work in a few muscle specific examples because it's a fascinating system to study mechanobiology. And as Ram mentioned, please feel free to speak up, to interrupt at any point in time. It's okay if we don't get through all the slides. You guys will have a PDF of this, but this is also sort of a more it's not a textbook area of science yet, right? So a lot of what we're going to be talking about are discoveries over the last 5-7 years. And so this is good material for more discussion and speculation. And so that's what I'm looking to get across to you guys today.
Ben, I'll just chime in quickly here. For the students, when you guys interrupt to ask questions, please remember to state your name, and then just to also, I think it might be helpful to Ben if you say where you're from. So if you say, I'm so-and-so from UNC Chapel Hill or UPenn or whatever, okay?
That would be wonderful. Yeah. Great. Okay, so the goals of today's lecture, the overall goal is really to just start building this appreciation that physical or mechanical cues can play a similarly important role as the much more widely understood chemical cues in regulating gene programs, and thus the regulation of cell development, cell behavior, or pathological progression. And we want to gain sort of a general sense of where the field stands on current mechanisms of nuclear mechanotransduction. And we'll talk more explicitly about what that means in a moment. And I want you guys to sort of appreciate also the cutting edge tools and approaches that are really required to investigate this widely untapped area of cell biology. So I want to start with a definition of terms. I'm sure you hear mechano added before everything. And so just so we're sort of on the same page here. So when I refer to mechanobiology, this is what we really sit at the interface of biology and engineering, the general study of how physical forces, changes in cell or tissue mechanics can contribute to development, physiology, and disease. When we put nuclear in front of that, and for this lecture, that's just when the above involves the nucleus. That I'm going to use that as a very general term today, and that is not the case for all mechanobiology. Mechanotransduction is more of the process. In the cellular context, this is the conversion of a physical force to a biochemical response. It can also be used interchangeably with mechanosignaling. And this is typically when an external force will elicit some intracellular response. But we'll talk about this here too today, even constrained to within the nucleus. And then mechanosensing, or a mechanosensor, is a protein or a cellular structure that initiates a mechanotransduction cascade, a molecule that senses that change in force and then is linked to eliciting a specific biochemical response. And one of the lessons I want you guys to come away with from today's lecture is that the nucleus itself is really a dynamic mechanosensing organelle capable of responding rapidly to changes in force. A little bit of background, you guys have heard more about this over the last couple of days, but just to reinforce some concepts, there are many different types of mechanical stimuli that a cell can sense and respond to. So we want to differentiate between them and understand them. And so just sort of refreshing mechanics 101, stress is defined as the force per unit of area, often expressed in terms of newtons per meter squared. There are a number of different types of stresses or forces that a cell experiences. This can involve shear stress, which is stress that acts parallel to the cell area. You can have a compressive force, like Ram just introduced, say due to a cytoskeletal polymerization force. This is more of a pushing force, or you can have tension or a pulling force, say a myosin motor pulling on an actin filament, or the forces associated with depolymerization of a cytoskeletal element. Strain is sometimes confused with tension, right? We think of things that are under tension as under strain, but strain can result from either compressive or tensile forces. Strain is a unitless term that just depicts the change in length relative to the original length. Right? So if you compress an element, that would be negative strain. If you extend an element, that would be an increase, a positive strain on that system. And stiffness is the relationship between stress over strain, often expressed in newtons per meter squared. This is this key material property, also expressed in pascals, which you'll see throughout today's talk. This is an example stress-strain relationship of a non-linear elastic material, which is a fairly good representation of a typical cell. The cytoskeleton within a cell is comprised of these cross-linked, semi-flexible polymeric filaments, like you just learned about. And as such, cells exhibit this very nonlinear viscoelastic behavior that includes this sort of power law stiffening of their elastic modulus with increasing strain. And so the slope of this relationship at any given point in time is the stiffness of the system. You can see strain stiffening here, where at higher strains you have a stiffer material. The other term you'll hear often is compliance, and compliance is the inverse of stiffness. As you become stiffer, you become less compliant. Okay, so I want to start by showing just some of the sort of prominent examples of nuclear mechanobiology in nature, and really just to illustrate how all cells are mechanosensitive, although for some, like muscle cells, it may play a larger role in their everyday biology, and how this nuclear mechanotransduction is really ubiquitously implicated in development in physiology and disease. And we're going to start here by showing some sort of gross cellular or organ level behaviors. And I hope by the end of the lecture, the goal is that you'll at least be able to generate some hypotheses or speculate on the molecular mechanisms that are underpinning that behavior. Not going to focus too much on disease today. We're going to focus more on the fundamentals, but it's important to appreciate that this mechanotransduction is strongly implicated in a number of prominent disease states, including atherosclerosis, fibrosis, asthma, heart failure, and cancer. So let's look at an example of a key role in development, and a rather famous example of mechanotransduction in action comes from Penn, where Dennis Disher's group, and maybe you guys are familiar with this paper at this point, has showed that the fate of a pluripotent stem cell can be decided simply by the stiffness of the substrate that stem cell is cultured on. And so here they took naive mesenchymal stem cells and they plated them on either soft, medium, or stiff matrices, mimicking the stiffnesses that are found in brain, muscle, or bone, respectively. And the stem cells that were plated on these soft matrices, they developed these neuronal markers, like this beta-3 tubulin stain here, where cells plated on the medium stiffnesses developed into muscle precursor cells, here expressing MyoD. And the cells plated on the very stiff surfaces, they would differentiate into bone cells, expressing here a bone marker. And so this was really the first demonstration that the matrix stiffness itself, upstream of any genetic or any chemical cue, can determine the cell fate, showing really a vital role of mechanotransduction in development and in stem cell differentiation. And this paper, I think, was a game changer, sort of set the stage. It's been cited, I think, 11,000 times to date since its publication. So this is probably setting the bar for the H-index for these authors. So let's look at a key role in sort of more physiological adaptation to stress. And so another one of my favorite mechanotransduction mechanisms is the remodeling of bone or tissue that can occur in response to changing external forces. And as a runner, one of the interesting examples of this to me is the adaptation that can occur during, say, barefoot running. And so in this study, these authors are looking at impact forces on a force plate from barefoot versus shod runners. What you can see is that the shod runners, they tend to first land with their heels. And this heel strike, this results in this large impact transient, shown here, this little spike, that some have linked to running injuries. Whereas a barefoot runner tends to land with more of a forefoot strike, as shown here, which promotes this decreased loading transient and a more even distribution of force over time during the cycle. This doesn't, of course, mean that you should just like kick off your shoes and start barefoot running, especially if you live in a city. A lot of these barefoot runners that are studied, they tended to start running from a very early age. And as such, the bone, the musculature in their feet have considerably remodeled from this force distribution to support that force distribution. And without that remodeling, you would certainly find yourself in a world of pain if you go out and try and run a barefoot mile from scratch. This last example is just sort of a key role in pathology. And so sensing and adapting to external forces, to the geometry of your surrounding environment, this is also vital for cell motility, shown here. And so this is an example from a 2016 paper from Jan Lemmerding's group, where breast cancer cells were challenged to migrate through this microfluidic device with these sort of seemingly impossible narrow constrictions. And as you can see from the video, the cells, not only could they make it through these tight squeezes, which you can imagine would be a pretty Herculean effort to break down their cytoskeletal structure and then reform it on the other side to allow that cell motility. And this is certainly an ability that could help a cancer cell, right, with its invasiveness or its ability to metastasize. You can also see this spilling out of this nuclear localized GFP signal that spills out as the cytoskeletal pressures on the nucleus as it squeezes through the constriction, rupture the nuclear membrane. But then you can see that sort of rapid reformation of the nuclear membrane, the resequestration of that GFP signal back into the nucleus, which would allow a cancer cell to navigate tight constrictions and continue on with its mission. Okay, so how do cells sort of accomplish these, what are really pretty amazing feats when you try and think about it? And so I want to start by looking at just the hierarchy of a typical mechanotransduction cascade. And so we start really with mechanosensing, right? This is where cells are testing their environment. The mechanosensors use force-induced modifications, conformational changes to transmit information about the cell's environment. And adhesion receptors, membrane proteins that link the cell to the external environment, are critical in this factor. And I believe you guys learned about adhesions in one of your recent lectures. Now, additional proteins and complexes link these mechanosensors often to the internal cytoskeleton, which may be involved in propagating that signal, right? So once you've sensed the signal, you have to propagate it. And this can be either through a chemically or a physically linked network. And so the extracellular matrix, the interconnected network of cytoskeletal elements play sort of central roles as force conduits in tissues. And then what you often have is signal integration at the nucleus. So that chemical or that physical signal can arrive at the nucleus, eventually altering its architecture, resulting in chromatin conformational changes or transcriptional patterns that are modulated to produce a response. And we'll spend quite a bit of time talking about this today. And then you have a cellular response. And this can occur in anywhere from microseconds to minutes and regulates a diversity of cell responses, including changes in shape, fate, motility, or growth. This is a cartoon example of common mechanotransduction pathways in a representative cell. And often a mechanotransduction response is mediated by several overlapping pathways and can be difficult to disentangle. And so here you see some common examples. We have mechanosensitive ion channels that can be activated due to the stretch of the plasma membrane, which can allow the permeation of calcium and other ions that serve as important second messengers. You can have regulatory molecules on the cell surface, like the glycoproteins that are found on the surface of endothelial cells, which have the ability to sense fluid shear stress, for example, to mediate mechanotransduction responses. You have cell-cell adhesion junctions that can facilitate inter cellular stress transmission via these desmosomes or adherens junctions. And you have cell matrix adhesion complexes. And these are where you guys learned about, you can probe the external environment, sense changes in the extracellular milieu or in the tissue strain or stiffness that's then propagated through the cytoskeleton to the nucleus. And the nucleus, again, is what's going to really serve to integrate these mechanical signals and accordingly modulate the transcriptional machinery to elicit a cellular response. But it's this last point, this how force modulates the transcriptional machinery, and how much of that is really due to force acting directly on the nucleus versus force activating upstream signaling cascades that then affect transcription through better understood pathways. This is still really an unknown and something that we're going to debate a little bit today.
A quick question.
Please.
This is Xiangjun from WashU, and I just want to know, how did the cell sense shear stress?
So there's others certainly within this CEMB that can answer this better than I can. My understanding is that the glycocalyx on the surface of endothelial cells undergoes conformational changes in response to shear stress, which activates signaling pathways, often involving Notch, Wnt, other classical canonical signaling pathways that can regulate cellular responses. But other than that, my level of understanding of that mechanism is fairly limited.
Oh, thanks.
Sure. Okay, so today we're going to focus again on how a mechanical force regulates gene expression. And this is sort of kind of a missing link in our understanding of cellular responses to mechanical signals, is how that signal is transduced to the nucleus, how it regulates gene expression programs. And so first I want to talk about some basic principles of genome organization and the regulation of gene expression that will be important for later concepts. And so first, let's just talk about this at the level of the chromatin. This is an EM image of a nucleus, and you can see these electron-dense regions, which are typically viewed as heterochromatin, which is, you see here, commonly associated with the nuclear periphery, although there are domains throughout the nucleoplasm. And the lighter colored is euchromatin. And so euchromatin is this sort of more easily accessible, active form of chromatin, where heterochromatin tends to have limited accessibility and is silent. So heterochromatin is this compacted, sort of closed form of chromatin that you can think about that's associated with gene silencing. And the activation to its more open form, this euchromatin form, that allows gene expression is often through the modification of these core histones by activator complexes that can contain epigenetic modifying enzymes that can, say, acetylate, phosphorylate, or methylate the histones themselves, or the DNA. Methylation also can play a role in this. And these modifications can be reversed by what are called repressor complexes, right? And these include, for example, HDACs, histone deacetylases, if you guys are familiar with that term, as well as DNA and histone methylases that can then silence gene expression by converting between these open and closed forms of chromatin. But gene regulation is certainly more complex than just euchromatin versus heterochromatin, right? So the genome organization in 3 dimensions is also critical. And so it's important to appreciate that the genome is organized into sort of higher order structures. We can start here at the left, where the DNA is tightly wound onto nucleosomes consisting of these histone complexes to form chromatin. These chromatin fibers can then be further formed into loops, which, for example, can place enhancers, say, right next to promoters to promote transcription in that region. These can be further condensed into what are known as TADs, or topologically associated domains. Okay, so regions of condensed chromatin that are organized into either transcriptionally active or transcriptionally inactive compartments containing many genes. And some of these TADs can be, sorry about the nomenclature, I didn't make it up. These can be associated with a nuclear lamina to form what are called lamina-associated domains. or LADs. We'll talk about the lamina in more detail in just a second. This is this layer of intermediate filaments that lines the inner nuclear envelope, okay? So now you have these whole, what are called chromosomal territories, okay? Regions of the genome that can involve multiple chromosomes into sort of similarly organized territories. And those themselves can intermingle with one another, which you see up here. And so these can form what are called sort of like transcriptional hotspots or transcription factories. So say, for example, you have a transcription factor sitting at the interface of multiple chromosome territories. This is why it's important to appreciate the 3D organization of the genome, right? Because if we think about the genome in sort of a linear fashion, right, just extending from one end to another, it can be very difficult to understand how a gene that's located over here can be simultaneously regulated by a transcription factor, or a transcription factor can regulate a gene that's located kilobases away in the genome, right? Because in their physical space in the genome, they might be right next to each other, okay? And so these form these sort of transcription factors, transcription factories that allow genes separated in sort of linear space on the genome to be co-regulated by activators or repressors in space and time. So when we're thinking about nuclear mechanobiology, please sort of keep this three-dimensional organization in mind. As mechanical forces that are generated by the cytoskeleton, they often have the effect of changing the shape of the nucleus. And you can begin to imagine the effect that that could have on this three-dimensional genome organization and the accessibility to different transcriptional factors and repressors. And so that's kind of the take home from this slide, is that, you know, much of gene regulation is really about accessibility, right, of different genomic regions to activators or repressors of gene expression, including the transcription factors, RNA polymerases, or enhancers.
Hello.
Please.
One question. I'm Gabriel from UPenn, I guess.
Hi, Gabriel.
My question is, like, have these genes that associate in TADs or LADs been shown to also correlate with like functions, as in
Yes.
all the genes in a given LAD work towards a certain function?
Yeah, that's there is certainly evidence of that throughout the literature. We'll get to some of that evidence in just a couple of slides, but that's sort of an emerging concept, is that you may functionally group genes into LADs or off LADs to regulate specific processes. And I'll show an example of that in just a moment. Okay, so another introductory slide sort of is about just how the cytoskeleton couples to this nucleus and to this three-dimensional genome organization. And so the primary linker between the cytoskeleton and the nucleus is the appropriately named link complex. Link complex stands for linkers of the nucleo and cytoskeleton. And the link complex sits on the outer nuclear membrane, spans through the paranuclear space, the inner nuclear membrane, and connects to chromatin. And there's two really critical protein families that make up the link complex. So this is the Nesprin family of proteins, which contain cache domains that span the outer nuclear membrane, extend into the cytosol, and which form direct interactions with each of the important cytoskeletal filament families, including actin, intermediate filaments, and microtubules that you guys just talked about. Those Nesprin proteins in the outer nuclear membranes connect with the Sun family of proteins that span the inner nuclear membrane. And these Sun proteins are in turn connected to this nuclear lamina, this meshwork of intermediate filaments in the internuclear space. And so the summary of this is this provides really a direct route for the transmission of physical signals from the cytoskeleton, which of course is connected to the cell membrane and the extracellular matrix, through the link complex to the nuclear lamina and to the chromatin that is associated with the nuclear lamina. So you have a route for force transmission from the outside of the cell to the DNA. Okay? And I want to focus a little bit more on these regions of these lamina-chromatin interactions for just a second, these lamina-associated domains, or LADs, as these are really important hotspots for nuclear mechanobiology. So this internuclear membrane and the nuclear lamina provide a scaffolding really for a vast array of important proteins and transcription factors. And the interactions between these elements are highly sensitive to the status of the nuclear lamina, which is dynamically regulated by force, okay? And so the lamina, again, is this very thin meshwork of intermediate filaments on the internal surface of the nuclear envelope. It's made up of the lamin proteins, lamin AC, B1, and B2, and this really provides the primary structural support for the nucleus, okay? So the majority of the structural support for the nucleus is given by intermediate filaments, at least in these mammalian cell types. The importance of the nuclear lamina is underscored by the fact that over 450 mutations to lamin A have been identified in patients and are known to give rise to 14 different human diseases, okay? So the regulation of lamin A is very important for diverse aspects of cell biology. A number of these, what are called laminopathies, also manifest as muscle pathology, which you can imagine in a highly contractile tissue, preserving the structural support of the nucleus may be particularly important to maintaining its function. Now, to come back to the question that I believe it was Gabriel that raised, LADs have been shown to play important roles in sequestering or allowing the active transcription of genes to regulate particular biological programs. And perhaps the best demonstration of this has been through in stem cell differentiation into more mature cell types. So if we look at the cartoon here on the left, in a stem cell, oftentimes genes that are specific for a particular lineage are located in LADs, sequestered at the nuclear membrane, and these tend to be transcriptionally silent, right? Whereas your housekeeping genes, your pluripotent genes, these are not tethered to the nuclear lamina, are more active, shown here in green, and that sort of defines the stem cell characteristics. But once you start moving towards lineage commitment, towards our committed progenitor cell, these lineage-specific genes come off of the nuclear lamina, allowing them to be accessed. And this can lead to the transcription of a lineage-specific gene program that helps drive differentiation into this fully differentiated cell type that has its unique sort of lineage-specific genes accessible. And so this is shown really beautifully here in some three-dimensional, I believe this is STED microscopy from Raj Jain's group, who's one of the CEMB members, looking at cardiomyocyte differentiation, where if you look in the embryonic stem cell, and here they're doing single molecule FISH to look at the titin gene. Titin is a key muscle-specific gene. Early in development, it is tightly associated with the nuclear lamina, and it's marked by this inactivating methylation mark, right? This repressive methylation mark shown here in blue, right? So this gene is presumably silenced. But as that cell differentiates towards a cardiomyocyte lineage, you see that that titin gene loci is untethered from the nuclear lamina. It loses this repressive mark, and it's now subject to active gene expression, further driving that cardiomyocyte specific fate decision. Okay? So I think LADs are really fascinating, and you can imagine how forces that would be imposed on the nucleus could affect chromatin interactions with the nuclear lamina. And this could form one mechanism for the regulation of gene expression. Okay, but we're going to talk sort of in more detail about the plausibility of that mechanism in just these next few slides. Okay, so this is our big question, right? Is how does force regulate gene expression? And again, not all mechanobiology involves nuclear regulation, but we know many of the responses we see in terms in response to force or changing mechanical environment must involve changes in gene expression. So how does this mechanistically occur? And we can think of this in terms of two really broad mechanisms, and then we'll build in some granularity shortly. And so the first is through sort of an indirect mechanism. This is force-dependent signal transduction, where say a cytosolic signaling cascade that could be initiated by forces at the cell periphery results in the activation or repression of transcription factors that regulate gene expression through sort of canonical modes. And this could include mechanosensitive channels or other force-dependent kinases, which we'll talk about in just a second. The other, which I think is sort of more fascinating right now, is this direct force transmission to the nucleus, where forces transmitted from the cytoskeleton push or pull physically on the nucleus. And this elicits some biochemical change that alters gene expression. It's important to note that these mechanisms are not mutually exclusive and likely each contribute, and even synergistically perhaps, to the mechanoregulation of gene expression. And so this cartoon shows some of that synergistic signaling of that indirect and direct mechanosignaling that here underlie the specification and the development of cardiac cells. And so on the left, right, you would have an indirect force-dependent signal transduction cascade, where matrix stress could activate integrin receptors, activate specific kinases at focal adhesions that could then drive transcription factor import into the nucleus and gene transcription. But in parallel, activation of those same focal adhesion complexes could lead to force transmission through cytoskeletal structures that, through altering chromatin remodeling or affecting nuclear import, can lead to similar transcriptional regulators that drive gene transcription. Okay, just showing the overlap between these pathways. I'm going to spend less time on these indirect mechanisms, but I just want to highlight this, and this will kind of reinforce some of the material here that you learned, say, from Liz Haswell, right? There's a prominent role of mechanosensitive ion channels in this indirect nuclear mechanotransduction, where a mechanical stimulus on the cell membrane changes the open probability of a channel permeable to second messenger ions that lead to downstream changes in signaling pathways that can increase or decrease gene expression. And many of these mechanosensitive channels there's a lot of fundamental biophysics and biology still being discovered about these. Some of the best studied are the TRP channels or the piezo channels, these massive channels that have now become somewhat famous in mechanobiology. And just a sort of a quick demonstration of their important role here in, again, the direction of cell fate. Here is where we're looking at the role of stem cells into differentiating into different neuronal cell types in the brain, whether glial cells or neuronal cells. And here the authors have knocked down piezo1, this mechanosensitive channel, and you can see that that drives an increase in the expression of glial cells marked by this marker GFAP, and a decrease in the expression of neuronal cells from this microtubule associated protein MAP2 expression. So just a quick demonstration of a role of mechanosensitive channels in determining cell fate. The other common sort of indirect mechanism is an activation of a kinase signaling cascade. And so here you have mechanosensitive proteins, often at the cell membrane, that deform, and their allosteric changes can alter downstream kinase signaling pathways to affect gene expression. And perhaps the two most famous of these are focal adhesion associated kinase, or FAK, as well as YAP-TAZ, which you guys may have already heard of. So a quick example of each of those. Focal adhesion kinase, again, is intimately associated with these focal adhesions. Its activity can be regulated by tissue stiffness, and it hits a number of downstream pathways that I'm sure most of you have heard about. Some of these, ERK, JUNK, RAC, RO, AKT, that regulate a large diversity of biological processes. YAP-TAZ is the other famous mechanosensitive transcriptional activator, I believe perhaps either first or at least mostly made famous through this Nature paper in 2011, where the authors plated cells on either stiff 40 kilopascal or soft surfaces, and found that on the stiff surfaces, YAP-TAZ, this transcriptional activator, was located in the nucleus, where in soft surfaces it was excluded from the nucleus. And this corresponded to a change in several of these critical genes. And we know that that gene expression change was due to YAP-TAZ, because if they knock down YAP-TAZ, this gene expression is no longer turned on, even on the stiff substrates. Okay, any questions on that? Just pause for a moment, and then before we really get into the material I want to spend a little bit more time on. Okay, great. Okay, so for the rest of the talk, I'm going to focus on the details of sort of more direct force transmission to the nucleus, where these interactions between the cytoskeleton and the nucleus transmit force to affect nuclear structure and function. And again, the important sort of take home of this is that it really positions the nucleus as a independent, dynamically mechanosensitive organelle. And I think as Ram sort of alluded to, this is a relatively new field, but more and more people are diving into it because there's a lot of fascinating territory to explore. So I just want to look at one sort of just to contextualize this example of sort of nuclear force forces applied to the nucleus through the cytoskeleton in a live primary cell. So here we're looking at the microtubule network at the top of a heart muscle cell. When that heart muscle cell is being stretched to mimic the way that heart muscle cells are stretched with each heartbeat during diastolic filling of the heart with blood. And you can actually make out one of the two muscle cell nuclei over here in sort of the top right. And you can see this nuclei becomes deformed and stretched with each stretch of the cardiomyocyte. And you can see these intimate associations of the microtubule network all along that outer nuclear membrane. That nucleus isn't stained with anything. That's just the microtubule mark, right? And we found that increasing the sort of the stiffness, the density of the microtubule network in heart failure can augment this sort of force transmission to the nucleus, which can have detrimental effects on nuclear integrity, on gene expression that can contribute to pathological progression. Let's talk more about sort of the typical generalized cytoskeletal forces that can be applied to the nucleus. And this is a representation of a standard cell on a stiff substrate. So you have sort of this apical and basal polarity. And cells attached to the extracellular matrix via integrins and other associated proteins that form focal adhesions, as you learned about. And then forces are generated either by strain of that tissue or by active cell contraction, are transmitted through the cytoskeleton, primarily by actin stress fibers, shown here in red, but also by intermediate filaments, shown in green, which often form sort of a scattered cage around the nucleus. And the tension within that cytoskeleton transmits forces to the nucleus, and that can initiate mechanotransduction responses. Now, this is the cytoskeletal architecture seen in many cells. It's important to notice to note some sort of context dependence here, right? So this is typically on a two-dimensional substrate, which is the most common system used to study mechanobiology. But it's important to remember that the cytoarchitecture can change considerably in three-dimensional environments or in different cell types. So for example, in the muscle cell here, there's a very dense microtubule network shown in yellow, right? And you can see that it's butting right up against the nucleus here at its long axis and sort of wrapping all around it. And intermediate filaments, which are shown here in pink, perhaps you can appreciate, instead of being this sort of chaotic cage, they actually resemble more ordered struts that kind of brace the nucleus from all sides. So just keep in your mind that the textbook cartoon of a cell line plated on glass or plastic may not reflect your cell type of interest in its native environment. And this is why a big emphasis of the CEMB is generating tools, 3D engineered tissue environments or organoid models that allow investigators to explore the role of forces in more native, more physiologically relevant contexts. Now we can get a sense of the forces that these cytoskeletal elements are applying to the nucleus by disrupting them systematically and observing what happens to nuclear shape. Right? And this is, again, particularly useful to do in a muscle cell, as because these cells are so rigidly structured, the cell does not actually change in size or shape with acute perturbations of these cytoskeletal elements. But the softer, more malleable nuclei will change size or shape when you alter the cytoskeletal forces that are imposed on it. And so actin fibers are believed to apply sort of tension to the nucleus, and you often see these what are called actin tan lines that normally cross over the apical surface of the nucleus and sort of squeeze it. So when you remove actomyosin tension, the nucleus will often round, right? Which indicates that typically there was some sort of squeezing force being applied by the actin network. Microtubules often apply compressive forces to the nucleus. So if you remove microtubules, often the nuclei will slightly elongate, right? Indicative that you've removed some compressive force that was constraining that nuclei. Now here, where we remove intermediate filaments, we actually see a collapse in nuclear volume. The nuclei becomes more wrinkled, and it actually decreases in volume. This may vary considerably based on the cytoskeletal architecture and the cell type you're looking at. But in multiple cell types now, the intermediate filaments have been shown to sort of protect the nuclei from excessive deformability and nuclear damage that could lead to this sort of crumpling phenotype. All right, so how do those forces actually change nuclear biology? And in the review you were assigned, the authors break this down into 3 potential mechanisms, really, for direct nuclear mechanotransduction that are reasonably well supported by evidence in the literature. The first of this is you can have a force-induced protein conformational change, okay? Where force applied or experienced by a nuclear protein could cause it to unfold, undergo a conformational change that could then change partners that it interacts with. It could allow post-translational modifications that can have downstream consequences within the nucleus. The second mechanism involves stretching of the nuclear envelope. We'll look at individual examples of each of these. And so if you stretch the nuclear envelope, inside the nuclear envelope, of course, you have the nuclear pore complex, which controls import and export of proteins and RNAs between the nuclear and cytoplasmic compartments. And so force could actually change the permeability or the accessibility of the nuclear pore complex to permit the entry of different transcription factors or small molecules or ions. The third mechanism is through chromatin modifications. And so force itself may be able to affect chromatin condensation or decondensation. And this can also permit or change the accessibility to chromatin modifying epigenetic enzymes, right? Which themselves then can affect that chromatin accessibility and gene transcription. And so we're now going to look at some of the literature and the best evidence that supports each of these mechanisms. But again, keep in mind that most of this is quite new, right? Arising from work over the last few years. So none of this is going to be considered really textbook science at this point. But I think that's a very exciting point to be in, right? I mean, you get to write the textbooks. And so this is a pretty cool field. All right, so one of the seminal papers to support that first mechanism, that force-induced conformational changes in a nuclear protein can regulate gene expression. And this group sought to answer sort of just a simple question. Is force on an isolated nucleus sufficient to regulate gene expression? So forget about focal adhesions, forget about transmission through the cytoskeleton. What happens if we just tug on the nuclear membrane? And so this work from Keith Burridge's lab at UNC was the first to really do that well in this 2016 paper. So it's a really cool experimental setup. So what they did is they used magnetic tweezers to apply force directly on isolated nuclei through the link complex component of Nesprin-1. So they took the magnetic tweezers to stimulate or move magnetic beads that were coated with an anti-Nesprin-1 antibody that then hooks up to the nucleus via the link complex. And then they could measure the bead displacement due to a known imparted force that's induced by the magnetic field, right? And that's what they're measuring here in B. They're looking at, with a given force placed on the bead, how much the bead displaces. And from Hooke's law, just F equals Kx, right? Force equals the spring constant times the displacement. If you know the displacement, you know the imposed force, you know how stiff your material is, right? And so what you can see is that with repeated force application, the displacement goes down, meaning that the nuclear stiffness is increasing. Okay, so what they're showing here is in real time, repeated deformation of the nuclear membrane is leading to stiffening of the nuclear membrane. And they went on to show the molecular mechanism underlying this. And this involves phosphorylation of a protein called emerin and remodeling of the nuclear lamina, which are just shown in sort of cartoon form here. So what the authors found is that force applied directly to the nuclear membrane leads to the rapid phosphorylation of emerin. And what this does is it increases the association of the nuclear lamina with the nuclear membrane. So you see more recruitment of lamin AC to the bead complex, right? Connected through the link complex to the bead than in the whole cell lysate, okay? And this happens quick, right? Within 1 minute, you're seeing more of this lamina association with the link complex. This phosphorylation of emerin, lamin recruitment to the link complex, they then showed was sufficient to regulate downstream gene expression on a rather rapid time scale, which changed several properties of cell morphology. So the point of this sort of seminal demonstration is just to establish that the nucleus is, again, really a rapidly mechanoresponsive organelle independent of anything that's going on in the cytoplasm. And that's a really important concept. Okay, so in that example, we have force that's sort of increasing this association of the link complex with the nuclear lamina. And this kind of dynamic regulation of the lamina, the effect this has on gene regulation, is really sort of a common theme emerging in nuclear mechanobiology. And so some important work from Dennis Disher's group in 2013 helped stimulate this interest in the nuclear lamina, when they showed here on the left that the nuclear stiffness is really determined by the ratio of lamin A to lamin B, these two lamin proteins, but that importantly, that lamin AB ratio, or that nuclear stiffness metric, correlated very well with tissue stiffness, sort of across cell biology. And so if you're looking at the plot here, right, they're showing this lamin A to B ratio on the y-axis, which you can take as nuclear stiffness, and this tissue microelasticity on the x-axis, and you can see this sort of power law relationship, okay, where the nucleus becomes stiffer in a stiffer tissue environment. We'll come back to that in a second. And they postulated that this would be a way to stabilize the nuclear lamina in stiffer tissues to protect from nuclear damage. And this idea of stabilizing the nucleus has been reinforced in several elegant studies, including here where, I can't pronounce the first author's names, unfortunately, they made these interesting chymographs of the nuclear envelope. So showing the change in nuclear envelope structure as a function of time from top to bottom. And what they found is that if they put cells on these sort of micro-patterned islands in a rectangular shape, right? So they impose some mechanical constraints from the environment, that this nuclear membrane was very stable over time. These are the gray traces here, right? There's very few fluctuations in the nuclear membrane. Whereas if they plated the same cells on a rounded surface that allowed the cells to round up, you see this considerable fluctuations in the nuclear membrane structure. And these differences in the stability of the nuclear envelope, they concluded were due to remodeling of the nuclear lamina, because if they knocked out lamin AC, even in these elongated cells that before were very stable, now the nuclear membrane fluctuations are all over the place, right? And the converse works as well, whereas if they overexpressed lamin AC in these rounded soft cells, that was sufficient to stabilize the nuclear structure. All right. So the take-homes of this work are that lamin A expression is really a key factor in stiffening and stabilizing the nucleus, and its expression seems to be driven by changes in tissue stiffness.
Hey Ben,
is that clear? Yeah.
Ram.
Ram, go ahead.
Have people looked at what, similar kind of experiments, but with like the lamin B receptor or any of the other inner nuclear envelope transmembrane proteins to which the lamina binds to? Like, is it just the lamin expression and assembly, or is it their anchoring to the nuclear envelope? I mean, I know farnesylation, et cetera, is involved, but there are a bunch of transmembrane proteins as well, right?
Yeah, so I'll show in the next slide one potential mechanism for this stabilization of the nuclear lamina. Absolutely, post-translational modifications are important as well, such as, like you referred to, the farnesylation of the nuclear lamina. This is what is compromised in progeria, as this form of lamin cannot be farnesylated, and which makes it extraordinarily stiff and this creates a number of problems, including premature aging. There's also, as you referred to, a number of transmembrane complexes that can regulate lamina stability. We won't be able to go through all of them today. Here I'll talk about the one potential mechanism that now has some additional validation for how forces could lead to lamina stiffening based again on this regulation of lamin A levels. But it's an important context to keep in mind that there are many different layers of regulation of the multiple lamin proteins and associated proteins that can influence lamina stability other than just expression levels of lamin A.
Thanks.
Okay, so we see this sort of how tissue stiffness seems to correlate with lamina stiffness and nuclear stability. And later the Disher group, with contribution from others within SEM, including our lab, helped to sort of propose a mechanism that could lead to stiffness-dependent lamina stabilization. And this is really where the force-induced conformational change of a nuclear protein comes in. So what Dennis's group found is that when you have an increase in extracellular matrix stiffness, say by increasing deposition of collagen 1, for example, this drives an increase in actomyosin tension, right, through focal adhesion signaling, as we talked about before. And this leads to increased force application through the link complex to the nuclear membrane and to the nuclear lamina. And that force-induced forces applied to the lamin proteins leads to a conformation of these intermediate filaments that hides potential phosphorylation sites. And when that force is removed, let's say when that tissue softens, you expose phosphorylation sites on lamin A, and they identified particular kinases that can target lamin A, and lamin A phosphorylation increases its solubility. So this more soluble form of lamin can then dissociate from this internuclear membrane space, becomes more nucleosolic, and is subject to degradation. So this is a mechanism whereby decreasing the forces of the nucleus leads to increased phosphorylation of lamin. More lamin moves into the nucleoplasm and is subject to degradation, leading to a compromise of the integrity of the nuclear lamina, or softening of the nuclear membrane. And this system works both ways, right? If you crank up the force applied to the nucleus, you protect those phosphorylation sites, you stabilize that nuclear lamina, you stabilize that nucleus in that stiffer mechanical environment. And they found that this is particularly important for stabilizing the nucleus as tissue stiffness increases during development, but also in certain disease states. And in a sort of a similar mechanism, similar roles of the nuclear lamina have been seen by Jan Lammerding's group at Cornell, where the lamina stiffness can protect from nuclear rupture. And again, this becomes particularly important in certain muscle diseases, where mutations in lamin A can compromise their incorporation into the lamina, lead to haploinsufficiency of lamin A. This compromises the lamina stiffness, and with the repeated stresses of muscle contraction, over time, this leads to nuclear rupture, DNA damage, and the progression of pathologies, including heart failure or muscle atrophy type diseases. How am I doing on time? Okay. Any questions there? All right, I'm going to skip this one because I want to focus on the other two prominent mechanisms in these last two slides. And so this is the idea of nuclear envelope stretching. I think this is probably the paper that shows this most elegantly as a potential mechanism of nuclear mechanotransduction. And so again, this involves stretching of the nuclear envelope, which can change the accessibility or permeability of the nuclear pore. And so this is just a snippet of their data here, where again, they're plating cells either on soft surfaces or on stiff surfaces, or they're using osmotic stress to change nuclear shape, right? And so what you see, we'll just stick with the stiff surfaces. You can see that the nuclei flatten, and this is quantified here on the left. And with that nuclear flattening, they noticed that there is increased transport of YAP into the nucleus, this important transcriptional activator, right? So They also did some elegant EM work with these cells on soft versus stiff substrates, and noted importantly that when you flatten that nucleus, which will apply, you can imagine, tension or stretch that nuclear membrane, this resulted in an increased size of the nuclear pore complex. And you can model how much this will change the permeability of the nuclear pore complex to transcriptional factors or transcriptional activators such as YAP. They also found that just poking on these cells with an atomic force microscope, applying local strain to the nucleus, was sufficient to increase nuclear pore complex accessibility and facilitate YAP import into the nucleus, where it can then regulate mechanotransduction responses. And so this is sort of their overall model here on the right, where exposure to a stiff mechanical environment through these connections between the cytoskeleton and the nucleus allows the focal adhesion forces to reach the nucleus, which leads to nuclear flattening, stretching nuclear pores, lowering the mechanical resistance to molecular transport, okay? It's important to note also other groups have also found that if you disrupt a link complex, or you disrupt the nuclear lamina, say through mutations again in lamin A, this can lead to infolding or wrinkling of the nuclear envelope. And you can imagine that nuclear pores in a stretched out sort of taut membrane are going to have greater permeability than nuclear pores in a collapsed sort of folded on top of each other membrane structure, okay? So certainly while more work needs to be done here, I think this mechanoregulation of the nuclear pore complex is an attractive model for nuclear mechanotransduction. There's also sort of more direct cytoskeletal interactions, particularly via microtubules and microtubule motors with the nuclear pore complex that may mediate force-induced conformational changes in the NPC that allow transcriptional factor shuttling and regulation of gene expression. And the last example I wanted to show today is maybe one of my favorites, but again, I think we should view this as like sort of a cool proof of concept, not necessarily an established biological principle. But this is one of the seemingly sort of most direct mechanisms of nuclear mechanotransduction, and that's chromatin stretching, okay? And this sort of proof of concept was provided in really a beautiful paper using clever approaches in 2016. And so the authors wanted to test whether direct stretching of chromatin itself is sufficient to turn on gene expression. And so they engineered what's called a bacterial artificial chromosome, or BAC, which contained multiple copies of this gene, it's DHFR, it doesn't matter, with multiple GFP tags spaced along that chromosome. So when you express this in cells, you see these sort of multiple little green puncta, right, which correspond to different regions of that same artificial chromosome. Then they use a technique called magnetic twisting cytometry, where you take magnetic beads that are coated with an RGD domain that hooks up to integrins, and you apply a magnetic field to twist those beads. So you're tugging on the integrins, which are going to transmit forces through the focal adhesions to the link complex to the nucleus. And then they monitor what happens to those GFP puncta and what happens to gene expression. And here's where they're looking at the distance between GFP puncta over time, right, as they twist on that cell. And you can see the beads are separating, the GFP puncta on the chromatin the chromatin is undergoing strain, right? The beads are increasing in distance from each other and then coming back closer together as they turn on or off. And they found this could be graded by the directionality of that force application, the magnitude of that force application, to really dynamically regulate chromatin strain, okay? And what they also show, which is super cool, right, is that this corresponded with a rapid induction in gene expression of this DHFR gene that was engineered into this artificial construct. So even within 5 seconds, or at least say 15 seconds, right, you can see that gene expression is increased upon this stretching of chromatin. And so while the significance of chromatin stretching definitely needs to be more extensively studied, particularly in endogenous genes and more endogenous contexts, right, computational modeling also supports this as a very plausible mechanism, right? So to say decondense a chromatin fiber of 30 nanometers, so to make it more active, you need only about 5 piconewtons of force. This is a small amount of force. It's the same amount required to, say, unfold talon at a focal adhesion. And the level of decondensation that's necessary to initiate transcription is also very small. So given that a gene loci right, will only expand maybe about twofold when you induce transcription through other artificial means, okay? So this is about just half the level of compaction that's present within a 30 nanometer chromatin fiber, okay? And so it's just, the take home of that is small forces are capable of decondensing chromatin to the extent that is sufficient to activate gene expression, right? So I think this is a very plausible mechanism. The other thing is that chromatin decondensation itself is capable of untethering gene loci from LADs at the nuclear periphery, okay? And so this may be a mechanism that synergistically works with lamina-associated domains to allow mechanical forces to decondense chromatin and make them accessible to different transcriptional activators or regulators. Okay, so I think I am out of time, and with that, I will wrap with just a few conclusions here that I think you guys can just read. Hopefully you got some of the take-homes from today's lecture, and I'm happy to answer any questions that you might have now in the remaining time, or also via email at any time after the fact.
Hello?
Yes.
Can you go back to the previous slide? So my question is, in the nucleus, are there any ways to have like localized stretching forces? Because it seems to me that this is very unlocalized, so it would just result in an overexpression of every gene, right? If you don't have a way of like forcing a specific chromatin to stretch.
Yeah, absolutely.
So
I mean, you can imagine a number of layers of local control over this mechanism, right? And so presumably how well a region of chromatin or the gene is associated or connected to the cytoskeleton is going to influence this, right? And this can be controlled by the positioning of the link complex and by the tethering of regions of chromosomes or chromatin to the nuclear lamina, right? This is going to provide a sort of more direct physical connection to the cytoskeletal forces that could influence chromosome stretch or chromatin stretching, right? And so, and there are a number of factors, including these acetylating enzymes, HDACs, and that can promote chromatin assembly and interaction, sorry, I should say chromatin association with the nuclear lamina or removed from the nuclear lamina. So control that tethering, right? And so that would be a way to accomplish sort of local control of chromatin stretching. Also with the directionality of the cytoskeletal force, or where the cytoskeletal force is being applied, right? Cytoskeletal forces are not going to be uniform throughout the entirety of the cell. And so if you have compressive forces applied by microtubules at one region of a cell, in one polarity of the cell, that could be a way to control genes that are closely associated with that application of force. So I think there's a lot of layers of regulation that could provide more specificity to this mechanism. Again, how biologically relevant it all is, it really still remains to be determined. Does that help answer your question?
Yeah, definitely. I guess because you mentioned the nuclear lamina and how it's bound to the cytoskeleton. And from my understanding, usually the chromatin that binds there is heterochromatin, so it's silent. And then this stretching leads to like activation. So it's, I guess they're like contradictory functions if these like nuclear located chromatin would be the one that be receiving the stretching.
Yeah. I think it's an interesting comment. And, you know, it's interesting to think about why the cell could use LADs, lamina-associated domains of chromatin, right, to sequester and silence particular genes that you would then want to be made available upon the application of mechanical stress, right? And so we think about this often in terms of in the heart. You know, the heart has to respond to mechanical stresses through hypertrophy, through growth, right? But you don't want to have constant heart growth, right? You want to be able to do this in the context of exercise, or this often happens with disease, for example. And so you could imagine sequestering particular genes that could regulate that response to stress in LADs, and then have them activated through these types of mechanisms by mechanical force to then make them accessible at that time of need. But again, that's very speculative, right? And sort of demands further investigation.
Sounds interesting.
Yeah, but I like this idea, right, of having like this little pool of genes that you kind of have set aside and normally silenced, but available on demand in the face of increasing mechanical stress.
That would be good. Probably.
Other questions, guys?
Hey Ben, this is Fabiola from UPenn.
Hi, Fabiola.
So I don't know if you mentioned it, but do cells like these cancer cells that have to be like more stiff and stuff, do they express more lamin A compared to softer cells?
That is that's actually a great question, and one that I'm not privy to the answer to right at this moment. You know, if we think back to this model put forth from Dennis Disher, it may depend on the tissue environment that the cancer cell exists in, right? And so, in order for the cancer cell to thrive in a particular tissue environment, it may have to regulate the integrity of the nuclear lamina to allow itself to maintain nuclear structure in a stiffer tissue environment. But you can also think about sort of the counter to that, where, you know, we showed this video of these cancer cells that are able to fully sort of break down their nucleus, and that will allow greater invasiveness, right? Because then breaking down their nuclear structure allows them to navigate incredibly tight constrictions. And so you could even think about this as almost a competitive advantage of the cancer cell. If they have lower lamina levels, they have more deformable nuclei, it would facilitate invasiveness and therefore metastasis. And so, again, I'm speculating here, but I just think it's sort of interesting biology to think about, and whether a cancer cell, through regulating nuclear integrity, if that gives it some advantage for proliferation or for invasiveness.
I guess it would be cool to have like a study in the future to track these sort of cells that are invasive at first to see like their lamin A levels, and then when they decide to like land their stuff and create a new tumor, like when the expression of the lamin A could go up to make them stiffer to compose the tumor.
Absolutely. I think that it's really an interesting question. I know that's a line of investigation that the Disher lab is pursuing, is how this is dynamically regulated in different cancer cell stages, for example. And there's probably more information known about that than I'm revealing right now. But yeah, I think it's really a fascinating area. And that's a great point.
Hey, Ben, this is Ram. There's something really I just thought occurred from that comment from question from Fabiola. I mean, I usually think of cancer cells as, you know, high mutation rates. There are sort of, you know, all kinds of problems. They're accumulating, you know, gene rearrangements, mutations, so forth, that sometimes can give them a further competitive edge, right? So now I'm wondering if this, what you just described, is true. If we assume that's true, I wonder if that would further sort of enhance, you know, contribute to nuclear changes, chromatin changes that would even promote that even more.
Yeah, no, I mean, I think it's a very good point, and it's something that's been speculated by both the Lemerding group and the Disher lab, right? Is that this nuclear rupture that occurs as cancer cells, you know, move through tight spaces, it does sort of promote DNA damage, which is associated with additional mutations that could sort of drive that tumorigenic potential even further. And so it's an interesting mechanism that I don't think we've thought about too much in terms of sort of the competitive advantage or tumorigenic potential of cancer cells. But certainly I think there's some interesting mechanobiology there, and DNA damage during these times of high nuclear stress has been definitely well demonstrated in cancer cells to occur, and so would be postulated to act through that kind of a mechanism.
That's cool.
Yeah. Any other questions, guys? These are great comments and points.
All right. If not, thank you, Ben.
All right.
Thanks all of you. You have some time to take a break before the lab session. I will not be able to attend the lab session because I have another meeting starting in an hour that will go through the lab session. So I will catch up with you guys tomorrow morning.
You can clap on Zoom. I see people are like, this is great. I'm so excited.
Like here, I can do thumbs up.
Oh, all right. I got to figure out how to do this. I should be more technologically advanced. Thank you guys for your attention. It was great. Thank you.
Thank you very much.